Good evening, everyone. This is a spontaneous, heavyweight scientific discussion. It's a follow up to a conversation that I'd previously had with Christie Grace, who has expertise in terms of the industry with regards to lipid nanoparticles and gene therapies and so on. And what had happened is that when I had spoken to her the last time, she highlighted some gaps that I realized were quite important. But I usually know that when something is important, it starts to get censored very, very quickly. And so this conversation ended up being censored quite heavily on some platforms. And what then happened is that as I continued to push the conversation, I realized that this is a very sensitive topic. Okay. She has done more work. She was due to speak with industry to raise awareness of what she's talking about. But I think there are some issues. So she'll be able to explain that in some more detail. But it indicates that this is something that probably shouldn't be ignored. Now, I can't say that it necessarily is a huge issue, but I suspect that this is something that nobody wants to talk about. So, but before we start, just a quick mention that coming up in another week, or frontline testing and treatment for post-COVID illnesses. That link, well, you'll have to check Eventbrite. The link is not in the description at the moment, but it will hopefully be there not long after if you're watching this after the live. But join us, register, and make sure you come and add your contribution to this important discussion. So without further ado, let's bring in Christie. Christie, thank you again for coming at short notice to share with us your insights and your knowledge. So firstly, can you just introduce yourself again?
Thanks for having me. My name is Christie Grace. I worked in biotech as a coordinator, project and process manager, using modified RNA to make recombinant proteins, things you probably would have seen similar to like a monoclonal antibody, and then working with researchers to design their ideas and bring those to clinical trial for researchers that have ideas on how to treat genetic disease and cancer. So lots of modified RNA work, did some with CRISPR and gene editing, and the area of recombinant proteins. So that's why I have an interest in the COVID vaccines, even though I didn't have anything to do with the manufacturing of those. But I worked in industry with teams designing product after product after product after product for different applications to see what some of these mechanisms are. And so the COVID vaccines really caused me some alarm with how they've been pushed forward to market.
I think this is an important discussion, Christie. And one of the things that I am trying to get my head around, and I've been following the science for a long period of time, and so I realized that this is something that really seems to evade the normal channels of understanding around the science. A lot of people are basing this on trust. They figure that if there were any problems, these would have been picked up. Okay. And it makes sense because you would trust the regulators and so on to pick up all these issues. Based on your expertise and what you see in this relatively niche field before the COVID pandemic, do you think regulators had any kind of expertise with assessing and understanding these products?
Yes and no. Yeah, mostly no. Some might have. When we speak to the background of people, because they're so new. When we talk about regulators, are we speaking to U.S. based or whatever country that they're in? We know that attenuated vaccines have been the focus of research and science, and that the tests that are done to check for harms or genotoxicity are based on attenuated vaccines. So it wasn't really in the working space of the people unless they had worked in biotech and then moved over to the FDA, or if it was their background, right? Like, we know there's a revolving door, but I think there's been a concern that even in the beginning, people were saying that, the COVID vaccines are a vaccine, and they're not. But they've been applying the same methodology and thought process to some of this. So I would say mostly not.
Yeah. So when we ask that very basic question, the mRNA technology, it has been argued back and forth for the longest while. It's not gene therapy per se, so they're not trying to implant a gene into DNA, but it's not ideally either in the context of a vaccine. Where does it fit?
That's just my opinion. Like, my opinion is it's not quite gene therapy, but it's not a vaccine either. It's something else. We know that attenuated vaccines introduce a weakened virus or part of a virus particle into the body, so it will generate an immune response. And the RNA is being introduced into cells to make cells to produce a foreign protein that they're not used to. But if we're just talking, I guess, semantics or definitions of words, they do matter in these contexts, because that's what the laws and the regulations are built around. And I don't think there has been adequate regulation for that. But gene therapy, like you said, usually you would, some of the work that I did was introducing a change in the human genome to bring about repair to a devastating disease that, say, impacts children, which is some of the work that I did, where children don't live a long time if they have a certain disease. So you want to change the genome, and those things are just starting to come to market. But yeah, I don't know that I could say that's gene therapy, but the fact that we're hearing that changes in the genome happened as a result of this is, it's a complicated area.
One of the criticisms when I first shared the discussion from people in industry was that mRNA does not enter the nucleus per se. So in theory, it can't actually have any impact on the DNA. And so the mRNA is in the cytoplasm, and it then makes the protein. What would you say to that?
We know it makes the protein. With regards to entering the nucleus, there's been, so that gets into a deeper, I guess, science discussion. The nucleus of our cells have protections to regulate what comes and goes out of the, what are called nuclear pores, and little holes. They're just channels, but it's really regulated. What someone makes as a product and what they intend it to do isn't always what the outcome is. So I guess there's a couple of things I would say here. We know there's linearized biotech plasmid, which has promoters on it. That is upwards of 200 billion pieces of it per dose. And we know the recent peer-reviewed publication by Dr. Speaker, Rose, and McKernan, and Taylor and Francis, they quantified that too, and they found levels of this DNA plasmid upwards of 400 sometimes the dose, which doesn't even matter. So RNA so we could go on a different, I guess, a stream talking about this. We know the DNA plasmid can enter the nucleus when a cell goes through its cell cycle called telophase. The mechanisms that protect and regulate what's entering the nucleus are temporarily kind of offline when a cell divides, so things can enter that point. So the DNA plasmid could get in there. The modified RNA could get in, but that would depend on if you want to talk about that for a couple of minutes, because the modified RNA could get into the nucleus as well.
Well, I think the point is that when we think about it, because as I said, we're speaking to a lay audience, so we don't want to get too heavy. But it did strike me that some of the concepts that you were speaking about there were quite relevant in the context of people just understanding what you said. So I'm just going to show, I'm just going to show a slide so that people understand what we're talking about. Here we have a normal human cell. Anything inside that gray, that purple cell wall is the cytoplasm. And then you have here the nuclear membrane, which protects the nucleus. And this is, again, protected space. So stuff that is in here doesn't easily get into the nucleus because it's protected. And so one of the arguments they've said is that mRNA, unlike even the AstraZeneca vaccine, which does go into the nucleus, all of the work happens outside here. In that context, they're arguing that it shouldn't therefore be a problem when you think of issues about DNA and adduction and so on. Is it just that there are more things that can get adducted, or is it that the only question is whether or not the mRNA gets transcribed into DNA, which that gets into the nucleus?
Right. All. So I think the answer to that would be all. There's been a study that showed reverse transcribed into DNA, correct? So where the modified RNA that's going into the arms can go back or can be changed into DNA. When we talk about the positively charged lipids that we discussed previously on, I think a month ago, that are inside of the lipid nanoparticle. So it's lipid nanoparticles, it's a blob, it's not a sphere, but inside of it are things like cholesterol, DSPC, it's called, ionizable lipid, modified RNA, thank you. And okay, so we know that DNA plasmid contamination is also in there. And part of that lipid nanoparticle, if you look at there's the RNA strands, the RNA has a negative charge to it in certain spots. So imagine a minus sign. And the ionizable lipid has a positive charge, and positive and negative attract. So the modified pieces of RNA have a surrounding kind of layer, and it's not perfect, of the positively charged ionizable lipids. And they're called ionizable because some of them already have that charge, the positively charged to them, when they're inside the LNP, the lipid nanoparticle. But when they get to a part of the cell, once they enter the cytoplasm that you talked about, what's called pH will be lower in a certain area, and they will gain a charge. So some of them will have more charge once they get in there. And why that's important is because we know DNA plasmid pieces are also inside that lipid nanoparticle, which means that the positively charged lipids are also attracted to the negative parts in the DNA plasmid in the same way they are attracted to the RNA.
Hold on a second. Yeah, so just a pause here. So you are saying, and yes, this is true, there's been a lot of discussions. The argument from industry is that the concentration or the levels are not relevant. But you're saying that, yes, not only the RNA strands, but DNA plasmids from the bacteria are sometimes inside these lipid nanoparticles. And the presumption is that even though they're there, when the lipid nanoparticle gets in the cell, these plasmids will get destroyed.
Nope.
But yeah, so this is now where the issue comes up. So you're saying, is it that they don't get destroyed, or we don't know?
There's different factors. So there's studies, and I could send them to you if you'd want. And I know they're posted on Twitter from a year ago, and I could retweet, where they injected mice with the same similar plasmids that made a protein, and they injected mice into the muscle with similar DNA plasmids that made a protein that glowed, and it made them easier to track. Or one time they injected plasmids into the muscle of mice or into their tail vein that made a really icky protein that made them sick. For the life of the animal, for almost 2 years, the protein kept getting made. And those weren't even inside of these kinds of lipid nanoparticles. And these studies were done a decade or two ago. So we know that in previous studies, if the DNA plasmid is in a circle, it starts off like a ring, a circle. If it's injected in, the animal body wasn't breaking it down, and this was tested multiple times in mice because they wanted to see if it would keep making the protein for a therapeutic purpose. Then we know that, and I'm sorry that I don't remember the names of the researchers of the study, found that modified RNA they're finding in the blood of people from the COVID vaccines up to 28 days, and it's not breaking down. So when we talk about the modified RNA for a second, we know it has methylpseudouridine on it, which is something that is meant to make it last longer. And with the positively charged lipids, like no one's come out and said this yet, and I think the studies need to be done, because the positively charged lipids, we know by Moderna's own study, are creating permanent, what is called covalent bonds. They're attached, like super glue. The ionizable lipids are attached to the modified RNA. The RNases, they're called, that are supposed to come in and break that down. But my concern is they're not able to access parts of the RNA to break it down because the lipids are kind of shielding it. So in the same way of the DNA plasmid, Kevin McKernan has made, he's come out on conferences and has stated, and so have others, that the way that the DNA plasmid is made, that there's parts of it that will take longer to break down. And those also have the positively charged lipids to it. So that what are called DNases, are those getting broken down? And things don't happen at the speed of light in the body.
So this is an important point because, as I said, I have been sharing some of these points and I get to see the pushback and what they say. One of the things that they would say from the pharmaceutical side, people who are defending the position, they would say, listen, whatever you may think, it has been injected into billions of people. If there was going to be a problem, even if we were to acknowledge, okay, maybe we could have done a little bit more testing, but it has been given to billions of people and they haven't seen any signals. Why should they be concerned?
Because they have seen signals, and the CDC has even reported it regarding myocarditis. We know there's a signal for that in young men. It's under the age of 30. So it was right on their website. We know that febrile seizures were another concern. And then we also know from the people that have spoken out and have injuries and have family members that have died that there are concerns. And those are only the people that we know about. Those are only the people that are coming forward. There's other people that we know from VAERS data that have injuries. But some are injuries that we don't know that'll happen or not in the future. We talk about data and guidelines. I could send you, it would take me a little bit just to quick grab it. There's a FDA CBER HHS guideline of 2020 on genotoxicity and genetic products. And they talk about DNA plasmids in this guidance document. So it was put out by the FDA. And they state clearly in there that there may be a concern regarding genotoxicity and integration into the genome, or the plasmids going latent, meaning they could be inside of the exosome that's kind of like hiding out in the nucleus for up to 20 years, and that there needs to be monitoring for up to 20 years. So I know you're talking about the signal, but the FDA has already put out guidance on this, and it feels like they didn't follow their own guidance sheet from January of 2020.
So when we think about that, then is why is it that as I reflected on this conversation we had the last time, and I was reflecting on what has happened this time, nobody else really seems to be talking about this? Is it that you are the only person who understands it, or is it that the other people in industry who understand this just don't care or don't think it's relevant?
There are multiple conversations going on right now. That's a great question. A lot of the focus is on DNA plasmid and SV40 and the DNA levels. and integration in the plasmids. With regards to industry, I'm shocked that when Moderna, if we're talking about the adducts, Dr. McMillan, are you talking about the adduct formation? I'm shocked that Moderna didn't proceed when their own study, they showed in Pacaratel 2021, when they mixed the ionizable lipids with their modified RNA. Of course, they knew that they are bound together because the positive lipids bind to the negative charges on the modified RNA, but the fact that they found covalent bonds forming, meaning an adduct, which is known to have drugs pulled from the market if adducts are found to form in certain levels with our human DNA, that they didn't advance and check to see if it was also happening in the human body. So when you ask, did they know? They knew that the lipids are creating adducts and bonding like super glue to the modified RNA inside the lipid nanoparticle. So for agencies, and this is where I'm trying to not get angry, and I'm pissed, for agencies to stock themselves with people who say they have PhDs and they're experts and they're MDs and they found those adducts within the lipid themselves, why no one set forth and tested it on human blood or animal blood to see if it was forming there using the same type of instruments, which is like mass spec. So yeah, I do have question whether or not people knew about it, because I can't just be one person sitting here on Twitter and they just didn't do anything about it, or they're just keeping their mouths shut. So it gets upsetting. So I think some of them it didn't think about it, but I have to assume someone did and didn't speak.
So you were going to be talking to regulators recently. Who did you want to share your recent information with? What were you hoping to gain from that?
I was going to place a request to speak for public comment to the CDC working group, rather. So they already had their working group meeting in September, and I had gotten word of that after the window had closed to present a slide deck on not just the adducts, but the other stuff that's in the paper regarding the improper genotoxicity test. But I have been waiting to present to the CDC working group, but I have to ask for it. So they didn't accept. They just haven't opened the possibility for people to speak because the US government right now is shut down.
So in effect, then the opportunity to try and raise awareness of this at that level, because once you put it in front of them, they can't say they didn't know.
It's on record.
It's on record.
Correct.
And so at the moment now, this is one of these unseen issues that is just in the background. And this is, as I said, when we spoke about this the first time, and this was what, a month and a half ago?
It feels
it's not long, but it's the first time that, I always ask challenging questions, but this time it was like, you need to stop talking about this stuff. This was really serious stuff. This time it got a different kind of response. So
what do you mean by that?
I don't want to say I don't want to say. All I can tell you is that the kind of censorship and pressure changed after that conversation
that we had about the ionizable lipids bonding with the ionizable nucleic acid. Lipids.
And what I realized is that even the challenge that was coming back about that discussion was not that you were wrong. It was that they didn't need to do it because regulators didn't ask for it. Do you see what I mean? And so I was saying then, I understand that the regulators didn't ask for it, and it was probably because they themselves are trying to get a handle and an understanding of the platform. But you who are using the platform know that these things are relevant. Should you, if you have been giving it to billions of people, is there an ethical responsibility? Not necessarily a legal responsibility, but is there an ethical responsibility to make sure that this is not relevant? That was the question that I was putting forward, because it became very clear that there was no way to say it shouldn't really be done. They just said that they didn't have to do it.
That's a terrible response, right? That they didn't have to do it. So it was, so that it has felt like the regulators are pointing fingers at the pharmaceutical company, and the pharmaceutical companies have been pointing fingers at the regulators. I don't know if you'd agree with that they have been saying, well, we didn't know. That was one response that came back, correct? Like in the recent CDC work group meeting, and I think Dr. Malone was asking some questions of the drug companies, and they gave a vague response. And I think Dr. Malone said, and I quote, fascinating. And with regards just to the DNA plasmid contamination, those tests have been there in the past for the levels, but it wasn't inside of a lipid nanoparticle. So that changes how it enters the cells and what it has access to compared to what that DNA would not have access to, right? So it's disturbing.
One of the concerns I have, and I recently again had an interaction with somebody, and I made the point that the platform, and some people will disagree, but I think that the principle of the platform to try and deliver either specific medication or even gene therapy in certain conditions offers tremendous potential, because we have no other way to manage certain diseases. Okay. And so the platform has great value to help to promote health and to relieve certain diseases. Okay. But I then went on to say is that I'm not sure, however, that this is necessarily the best platform for vaccines. That's a different conversation. And I'm saying that I can understand that it was used in an emergency, and potentially because we know we can get an immune response if you had some other weird and wonderful virus or something that turned up for which you really needed to do something in a short space of time, and you just needed a quick way of doing it, it could potentially be used again. The problem we've got now is that they are extrapolating the pandemic into almost every vaccine. And so, therefore, if that is happening, you definitely need to do the full, thorough, appropriate testing. Is that reasonable?
Yes.
And so, within that framework, when we look now, is that how can I'm still trying to get my head around it. I can see with regards to COVID, the emergency situation. But we are now developing vaccines for influenza that are mRNA, for RSV, for. Is it that they have still not done this kind of adduct formation question research? I'm still trying to get my head around that.
Can we look at the recent paper I just submitted to preprint to kind of answer that question, and what the current regulatory guidelines are and how they are not mechanicalistically capable of doing the tests? When you said, what, thanks. So, yeah, and thank you again for reaching out to me. For anyone watching, I was waking up this morning having coffee. I'm in Central Time Zone. Dr. McMillan, I think, is 5 or 6 hours ahead of me, and he asked if I would come on today and speak. After, if I don't have $4,000, I think it's $4,000 to $5,000 to submit any of these papers for peer review, because I know there's question, well, it's not peer reviewed, therefore it's not relevant. That's not true. I don't think that's true. So after you and I had spoken about the adducts, and we were talking about testing and how do you test and what's happening right now in the government and the regulatory agencies around the world, I started to look for documents that were at least public. Okay, exactly what are they testing? Because the majority of people speaking have said they didn't do any genotoxicity testing, and I was thinking that too, and I didn't really go and look and see exactly what that meant. And Pfizer has been the focus on most of the injuries because of SV40. So it's been Pfizer, SV40, Pfizer, SV40. So I went after Moderna, and not just the United States documents. I went and looked at the EMA in the UK, and what the Therapeutic Goods Association, the TGA, And then I looked at Switzerland too. So I pulled a few documents in USA for Moderna, and what I found was pretty upsetting. And so I put it in a paper. And the first paper I looked at was Moderna. And this is Moderna's safety data sheet. And it talks about the drug substance. And I was curious if they did anything for genotoxicity testing. And if we go down and look, and I'm going to look on my screen at the same thing on my different computer here. The first thing that it says is what? Because they actually have carcinogenicity listed too. That's interesting. So genotoxicity, this is the Moderna basic safety data sheet that says that no data is available for this product or other chemically similar mRNA vaccines using the same LNP platform. And that we're going to get into, I think, in a second here. And then it also states that for the novel lipid in the mRNA product in vitro, meaning in the lab, not in an animal or in a living organism, it says that they used what's called the bacterial reverse mutation assay, the Ames test, and what's called the micronucleus test. That's for a PBMC, for peripheral human lymphocytes, and it shows that there's negative. And so this is where I paused and I thought I need to dig deeper into this because this was the first sheet that I pulled up. So I didn't know if you wanted me to explain just briefly, like, so what they did was, so they said the novel lipid. So I had to go to other countries' paperwork to find exactly what they meant by the novel lipid, because they are not talking about the whole product. They're not talking about the whole bubble that you showed earlier with the lipid nanoparticle with everything inside of it, the modified RNA and the other lipids. They're talking about the novel lipid, meaning the ionizable lipid with a charge. And what they did was, they put it alongside like E. coli, and there's another bacteria that they use. And these tests are what are called legacy tests. They've been around a long time and they've been used on multiple drugs to test to see if the drug would enter the bacteria itself and interact with the genetic material inside the bacteria. And then for the micronucleus test is the same. They kind of mix it up, and it's more science than that, but they'll take the drug substance, like Zantac, for instance. That's one to talk about that later got pulled from the market because they found adducts were forming after the fact. So they take a PBMC from an animal, or sometimes a human, but usually an animal, and they see if the drug is going to enter the PBMC, which is our lymphocytes. Those are immune cells, and if it will do anything inside of it or interact with the DNA in its own nucleus. And if it does, that's supposed to trigger a red flag to then do more tests. Well, the thing about the bacteria that's used in a nutshell, that's explained in the paper that I have listed, linked up to you, or I should be in a second here, is that the pores in the bacteria are smaller than the size of the lipid itself. So therefore it can't get in. Not going to get in anyway. So that's maybe a good thing. Maybe that's a good thing. That's not getting in. So I just want to call that out. It can't get inside. But they only use the positively charged lipid. So for the lymphocyte, that's called the PBMC. So they list a negative result for genotoxicity. But I don't have any props, but they only used a positively charged lipid. The positively charged lipid can't get inside because the mechanisms aren't there to do it. It's like going to the airport and waving a metal detector wand over a suitcase, not opening it up and saying, see, it's all good. There's no danger here. The lipid nanoparticle can get inside of a cell, but they didn't test that. They said for novel lipid in the mRNA product, and they don't call out exactly what part of it was. That's where I had to go to other country data to find out that they didn't test the full LNP on the what's called the micronucleus test. They just tested the positively charged lipid. So it can't mechanically, it can't get inside. So they said, see, look, it's fine, but it can't even get inside anyway.
So hold on there. So you are saying And I think for people to understand this, because I just realized what you were making the point of. So what you are saying is that they tested with, firstly, they tested these ionizable lipids with bacteria, but they're too big to get through the pores in the bacteria anyway. So it
That's
would be
good.
negative.
Right.
Then they tested these with the lymphocytes,
Hmm.
but these can't get into the lymphocytes in any case
Not very
because
well.
they're not very well. But what they should have been testing was whether or not the whole lipid nanoparticle
Yeah.
gets into the lymphocyte. And
Yeah.
once it's in, what happens with these ionizable lipids? You know, something struck me. A paper had just come out recently, and what they were finding is that the immune response to the ionizable lipids on its own was one thing, but even between the Moderna and the Pfizer, because they use different ionizable lipids, they get a different immune response. And so there is no clear baseline to operate from in understanding how they have the effects. They would have to test each one separately.
And the whole thing, but the
With the
interest
whole. Mhm.
right, the interesting thing about that too is that the PBMC, they're called, they used resting PBMCs, meaning not activated, meaning the immune system isn't active. The cells aren't active. They're not, the immune cells like lymphocytes kind of, our PBMCs are part of our immune system when our body gets threatened by a virus and they become active. They didn't really test that. And when gosh, that comes again, other conversations. What happens when the immune cells are active? And so studies have shown, which are in the paper, they have done tests on LNPs to see if they'll readily get into these PBMCs. They don't really anyway. So the LNP, it's kind of hard to get into these specifically unless the immune system is activated. So what if you shoot up somebody with an active autoimmune disorder? What if you put this into somebody who already has an immune system response going where these PBMCs aren't activated? They're kind of like sleeping. They're not doing anything. So the full LNP wasn't tested. The PBMC is probably not even the correct cell to even test against the LNP in this anyways. They only chose to test the ionizable lipid, just that one part, and it can't invaginate. It can't phagocytose. Like if anyone's seen photos, and I'm like moving my hands around here, but the lipid nanoparticle hits the outside of the cell membrane, and then it kind of pulls it in like a bubble, and then it goes in and it goes to different parts of the cell. The ionizable lipid isn't capable of doing that, but the ionizable lipid does not represent the product that was injected into people. Therefore, a completely irrelevant test was done where they say C negative result. It didn't harm the genome at all.
So when we look at the European Medicines Agency, and this
That's
is
a good
this
one.
is this is their similar to I guess what Moderna put in, and I'll pull down to what you showed me as I'd highlighted it somewhere if it will just flag up. When it comes to genotoxicity, here it is, genotoxic studies.
Mhm.
They tested it again with bacteria. Well, explain what the relevance of this.
Thanks. So the other document that we looked at by Moderna just said novel lipid. So I wanted to confirm they were saying that lipid that has a positive charge to it called an ionizable lipid, or how it can gain a charge when it enters the cell. So that's why I started looking at other data to see exactly what they're talking about. So again, this has been Moderna and the SM102. So the genotoxicity studies, it shows that they used this. So they're confirming, and this is on your side of the pond for the EU. So Moderna calls it out a little bit more here in the document, saying, yes, they use the ionizable lipid with the bacteria, and that they call out the strains that they used as well, that they used E. coli, and then the other bacteria to see if it would pass through the pores of the bacteria and get in at its own genetic data. And this is important too, because we as humans, we're not just human cells, we're 2 to 1. It used to be 3 to 1, or do we think 2 to 1 bacteria to human cells? Like, we're walking around with a lot of bacteria in us. So that's why it's important to test some of those to see if a drug can harm it. But then they also call out that SM-102, that's the ionizable lipid, was used in this micronucleus test using these PBMCs, and they call out lymphocyte. And they said that was negative. So again, they didn't use the whole product. They didn't use the full LNP against those red blood cells, or the peripheral blood cell, or lymphocytes rather. They just used that one little lipid piece, which is not showing how things will work in the human body when you inject it in. It's not representative of the action of the drug itself on cells or any interaction. And then what gets more interesting is in this document, they list the Zika mRNA, which sounded crazy to me. So they said, hey, look at when we made this drug for Zika. What was it back in, do you know the years when that was, Dr. McMillan? Was that back in like
Yeah, I'm trying to remember. Was it between 2010, 2015?
Somewhere around there, 2017, and
Yeah.
it never went to clinical trial. It never went to clinical trial. The drug for Zika never went to clinical trial. It was never approved. It was never FDA approved. It was never approved for use. And yet, I think there might have been an emergency authorization on it, on that Zika drug. So Moderna is saying, here, look at this drug that we made this one time that never went to clinical trial. We're saying that we did toxicity study. But when I pulled up the toxicity study again for the Zika, they were just using that PBMC. And then when you dig deeper into the data, the majority of what they did was biodistribution, where they showed it goes everywhere. Back in that time, when they were telling us it just stayed in the arm. And so this is even makes it a little bit more, what's the word I'm looking for other than concerning, that they said it just stayed in the arm. You know, they submitted the biodistribution data for the Zika study saying, or the Zika drug rather, instead of the COVID drug, but they didn't do full tests on that either.
So one of the problems with these conversations, Christie, is that the industry, regulators, as you can see what's happening with the CDC, they've gotten quite a hammering during the pandemic. Even doctors, trust has dropped 30%. Everybody involved in healthcare during the pandemic seems to have been absolutely hammered. When you raise these questions, you're hammering them harder. Should you just leave it alone? What is the best thing to do?
People should hammer harder. I should have more. Thank you for you having me on. So the hope is that, aside from tagging people on Twitter who will probably never see it, we need to immediately do proper tests. We can't keep on. These drugs were approved based on improper genotoxicity tests to say that they got negative results, when it won't even activate the cell, that part of the cell that needs to anyway. So it's moot. So we need more people, but it needs to be in a way that is less ad hominem. Take the blinders down. Know that it's not always a fight, and to, I guess, lower the tension, because people have lost lives and people have gotten sick and people have been injured. So a lot of people are upset. I've been upset. Just to lower the temperature down when we have these conversations, because right now, like you said, no one wants to even have the conversation. So hopefully, if you know anyone on your side of the ocean who you could show this to, I guess, we need to change the laws. So my hope was asking to present at the CDC working group. That was my hope, to show this data, to say we need to now make changes. We also need to go back and do the necessary genotoxicity tests, because these are not the right tests. So how do you change law? You can change law via petition, right? You can have a petition in the UK. Sorry, I didn't mean to interrupt.
It just struck me as you said that, because there are a lot of people who will be watching this who are not necessarily science-based. they don't understand necessarily the full ramifications of these kinds of discussions. But you just said something there that struck me. Here is the paper. It's in a preprint server. It's here, the author, Christie Grace, the abstract. Now, what people can do is that there is a link in the description. They can go to the preprint server, they can download the paper, and then they can share it. Okay? So it's kind of like now I'm empowering people to make sure that the right people can't say they didn't know. So now think about this a minute, Christy. So people are listening now. There is a link in the description for them to click on that link or copy it in. They can get and download the paper, or they can share the link in an email. Who do they need to send this to raise awareness, where that person can't say, I was not aware of even this question?
That depends on what country you live in,
Yeah,
correct?
just generally. Yeah, just generally. So just imagine, is it their MP? Is it their
Oh, is it Rupert? Rupert, what's his last name? Rupert
with,
Lowe. I'm trying to think of the MPs or the people in your country.
no, don't worry about
The
not just
origin.
my country. I'm just saying
Right.
anywhere in the world. What would you advise people if they are sharing this information and you want it to make an impact, where almost any country, this is put in front of their respective regulators? Who would that be? If I dropped you in India, if I put you in some anywhere in the world, Argentina, who should get this information?
The politicians and the regulatory agencies. The pharmaceutical companies, I mean, you could send them just to say, I sent that to you, but they could say, I didn't open it. But I know in the United States, our politicians like Thomas Massey and Rep. Green, like I had sent them some of the data, and the CDC, the FDA, HHS. So I'm sure they get inundated with it, but if enough people send something in, that might make an impact, where it's not just one email in a mountain of other emails coming in.
And so when you think about it, then they send the emails. And so let's imagine thousands of people watch this. They all get the email link. They send it to the various politicians and so on. What is it that they need to tell these people to do?
Do the proper tests and look at changing the regulatory rules, because that's what people have been using as an excuse. The regulation, right? They've been saying, we did what the FDA told us to do. We did what we were told to do. So the rules have to change.
Mm-hmm. So the rules need to be changed. They need to be challenged. Now, the question would be, so these people get these emails and they're getting them all the time. Their question is, what's the big deal anyway? If this is relevant, how will it manifest in terms of disease?
Then we have past precedent to look at. And that's why I brought up Zantac. The drugs in the past that have been removed from market that were found to cause adducts with the human genome, which means they're shown to cause a change and be a genotoxicity. Like Zantac was one. So I guess there are drugs that set precedent that were looked into and some were removed from the market. So I guess mentioning some of those drugs that were removed because they were found to cause adducts after the fact. It was after they got to market. That showed that these tests were ineffective in even those drug instances because, gosh, I'm going off on a tangent here. Like when you take a drug, it's metabolized by your body and it can form what are called intermediates. Kind of like if anyone drinks alcohol here, I can't drink beer, wish I could, I can't metabolize it properly, I get sick. But when you drink whiskey, if you drink beer, that is broken down into other substances. And what has been found in the past that these tests don't show, even with other drugs, is that things can change in the body and then adducts can form, and it can be a cancer concern. So we know Zantac. We know there's other drugs that have done this. So I think that's the concern. That not knowing is not an excuse to not do the proper test to check what the risk is. Because 5 billion people, and if, like you said, it's going to keep rolling into now what the influenza vaccine is going to be changed to this, if the HPV is going to be changed to this platform, there's great concern here. It's one thing to have something rolled out when there was an assumption of an emergency. That's not there anymore. Even if we don't agree that there was an emergency, it's time to pause it.
Yeah, I think this is the point, is that in effect, there is what you call grandfathering of the technology. This is what, when I spoke with Susan Langlois, I should try and reach out to her again. So she was in industry and retired, and she highlighted that important point, that what can easily happen is that just as you had the pandemic, if it gets put across that, this was given to 5 billion people, it's safe, there's no concern, let's move on. Let's just make vaccines with this because it's fine. And so you grandfather, this is how it works, is that you grandfather the technology so you're able to skip a generation of testing. And you're able to then just bring it to market, which is exactly as she had predicted. Instead of going back and doing the necessary studies. So say, for instance, I know that there were supposed to have been parallel animal studies that were started and then the animals were killed late. So we don't know what are the implications of the ongoing, especially with boosters and so on. All of these things happened in the context of the emergency. But you're right. The emergency has passed. Therefore, there is a responsibility to go back and make sure all the appropriate tests were done. And a useful historical analogy, is, I have to be careful with the word because it triggers so many things, but thalidomide. The United States was saved from the impact of thalidomide because of one woman. I've forgotten her name now. And she just kept on insisting on testing to be done where there were gaps. And she saved a huge proportion of the United States because only a few people who could get it privately actually got thalidomide. And in the same way, history teaches us that skipping the testing will only come back to bite us. And some farmer people may listen to this. And here is what I said to one of them. The technology has use. You worked in the industry. You understand the value of the technology. Are you willing to let the whole platform be destroyed in the eyes of the public because people want to make money off vaccines? That's really the question.
That was an excellent sentence. That was a great way to put that. Yeah, she's right. And there's been other precedent too when you speak to fast tracking. There's been data on the FDA. There's a study that was done, and it was more of looking at meta-analysis of drugs that have been springboarded off of other approved drugs, where there was actually approved drug, where the drug for Zika virus was never approved. It never even hit clinical trial, but that was used in the EA documents, or the EU rather, to support a lack of need to do further tests, when those tests weren't even fully done either. It never hit clinical trial, but there have been drugs that have been approved that were based off of other approved drugs. And then it was found out later that the foundational drug that all the other drugs springboarded off got recalled for harms. And then this set of drugs are still floating around out there. So we're in the same situation right now, except the platform was never fully approved for prime time, and now we are racing to a very concerning area. And you're right about trust.
Yeah. When you look at, as we come to the close, Christie, I want you, because I'm looking at your abstract here.
Do I need to clean it up? It was
Huh?
a lot. Sorry, I was judging myself on that. I said, do I need to clean it up?
No. My question is, when you look at just the abstract, what would you say is the most important piece of this abstract? Because it's a lot of information. What do you think is the most relevant?
The one sentence, however, these assays have the potential to be structurally and mechanistically incapable of detecting relevant risks.
Where is that?
Maybe about 4-5 sentences into the abstract where it says these assays have the potential. I also had to word it in a different way because I did get a kickback from preprint.org that didn't want my initial paper in there, which had a bit stronger language. So this is a little bit like neutral language. This is not my first draft of this, but yep, the assays that are currently used, the tests that are currently used by regulators all over the world are not structurally or mechanistically capable of assessing risk for these drugs.
And is this, and because one of the things, as I say, I'm very aware of the difference in terms of platforms. So think about this carefully. When you think about the work that you had been doing in the past with mRNA technology, are you saying that even then the testing of that technology before COVID vaccine still was not structurally or mechanistically capable of detecting risks?
Correct. And
even before?
Yeah, and to kind of go off of that too, the work that I did on teams that had really large hands in bringing some things to clinical trial to help kids that, when I say kids, I'm not trying to like plea for like an emotional response there either. I just want to clear that up because I know that happens sometimes. Adults too. The research and the treatments that are being designed and were being designed by researchers that wanted to bring something to market to help people with genetic disease and cancer, the demographic, the population that those drugs were being made for are people that had shortened life and suffering. That was their plan A. Shortened life. Not suffering free. I mean, we all suffer, but they're devastating diseases. So those drugs, as you said, this platform was for people that are dying young, for people that are not able to walk, the people who are not able to enjoy life. And so they came with risk. Where plan B was getting help and seeing these devastating symptoms disappear or be lowered in exchange for risk, and the regulatory agencies and the rules involved still are not adequate.
That is probably one of the most important points with regards to this. When these products were being developed, they were primarily focused on trying to extend the life and the quality of life of people who were already sick.
Right.
They already had either disease, whether it's cancer, or genetic disease, for which there were limited therapeutic options. And so therefore you're in a position to try more stuff, higher risk stuff, in order to bring benefit. And that's the beauty of medicine. This is why I said I know the platform has great value. But what seems to have happened here is that there was a window around the pandemic. And I remember when I spoke with Nicholas, oh goodness, I spoke with somebody in industry who worked with vaccines, and he said at the time in May of 2020, they thought there is no way that mRNA technology will ever get approved because there are so many hurdles to jump through. It's just not not possible. And then when it was fast tracked, that changed everything. And so suddenly, and I think this is the important bit, is that the technology, I still argue, I don't see from my angle the fact that in order to make a foreign protein to trigger the immune system, the lipid nanoparticle as a vaccine has to make the cell suppress interferon. So there has to be cellular interferon suppression in order to make this foreign protein for the immune system to attack. Fundamentally, that just doesn't seem right. And so my question still is that I am genuinely not sure, and I don't think I should be censored for this opinion. I am just genuinely not sure scientifically that the platform should be used, except in an absolute emergency, for vaccines.
Right. Unhealthy people. Like if it were Ebola, like
Yeah.
if we think about devastating
Yeah, so
things like
imagine
that.
you have Ebola, it's spreading rapidly. Granted, it doesn't spread that rapidly, but if you imagine a variant of it that was just devastating, for which if you could make a protein that would protect the person who was injected with it, yeah, I could see that in an emergency. That's what we have proved, that in an emergency, we have the potential to use this as a technology to help us to do things quickly.
I
But
think
what
that'll be
they're
also, sorry, go ahead.
hmm?
I
Yeah,
was
yeah, but
also
what
going to
they're
say,
doing now is transitioning this
yeah,
into everything without the appropriate investigations.
I was going to say that because I knew someone was going to bring it up. Like I know it's that it didn't work anyway. Like
What
the
didn't
COVID,
work?
that there's studies that have been conflicting whether or not the production of the spike protein in the human body didn't help anyway.
Yeah,
That they were,
well,
it wasn't effective anyway. But like,
yeah.
yeah, if we're playing devil's advocate and
If we're
saying
playing
we've got
devil's advocate,
this
so
population suffering,
what I can say quite honestly about the platform, and is that I know that it can trigger the immune response to target a specific protein. That I think is without question. My point is that I think in any case they picked the wrong disease. COVID is a disease where the immune system kills you for severe COVID. So it's like I just recently did the presentation. I compare severe COVID to rheumatic fever. It's the immune response that does the damage. So, how in the world are you going to get that perfect balance of immunity where it reacts just to the virus and doesn't overreact and destroy the person? That's that's fundamentally why I think this is the wrong disease, but that's a separate issue. The point being is that the technology has shown that it can make the immune system make a protein and trigger an immune response. In an absolute emergency, I would think, yeah, if that's the fastest way we can get to market with potentially an urgent vaccine whilst we work on others, then that's reasonable. But what is happening now to me, in my mind, doesn't seem right.
so we've reached the unethical space. If we weren't in the unethical space before, uh, and people have differing opinions in 2020, we are absolutely in the unethical space now to continue having this and adjacent products in the market.
So, Christy, as we close out, um, as I before we close out, before I capture her last words, just a reminder for people who have just joined us, what we are proposing to do is that in the description is the link to this paper. It's a preprint, but it has all the information that Christy is talking about. Remember, it's not whether or not Christy is right, it's whether or not these tests have actually been done. But most people have no idea that these tests should even be done. So, what it means is that your responsibility is simple: get the link from the description, download the paper, or just share the link. You send an email to whomever you think in your country is relevant to know about this information. It may be about 4 or 5 people. One may be your MP or whichever politician is in your area. 2 may be to the scientific regulatory organization in your country. And you may send it to your prime minister, whoever. The point is that if everyone acts and takes this information and shares it around, we cannot have the excuse in the future that we didn't know. Because at the end of the day, we just need the tests done. And if everything is fine, no problem. So what are your final thoughts, Christie?
It's blowing my mind that I have to think about this stuff, or anyone else has to think about this stuff. I think it's terrible that people aren't getting help who have COVID vaccine injuries. People who even have long COVID. People have lost family members. So I just wanted to say that I appreciate people who have spoken out like yourself and the risks that came with that, because there are not enough voices speaking out for the larger number of people that need help. And I just wanted to say thanks. I think that was it. Just that, yeah, I hope making some contacts with some regulatory agencies will help, because that one sentence there that you highlighted is the excuse that they've been using, that we did what we were told.
Yeah.
So just
Thank
say
you.
thanks. Yeah.
Yeah. Really appreciate it. Again, Christie, appreciate the short notice. Appreciate you doing the work. Appreciate all that you have done to raise awareness about this. And as we said, at the end of the day, if there is no problem, that's even better news. And that will help pharma, because they will be able to say, look, we have done all the necessary tests. Everything is fine. There is no concern. And everybody will be happy. But to not do it, especially when they are now aware that it hasn't been done, that borders on negligence. And for that, I don't think that there is a good excuse. So thank you again, Christie. I'll ask you to wait with me for the outro, but just as a final reminder to people, coming up next week is our conference looking at frontline testing and treatment for post-COVID illnesses. Join us. There will be a link. I'll continue to talk about it throughout the week, but just look out for it, 6 p.m. UK time next week, Saturday. And that's where we will be in terms of understanding what happens next. So again, thank you, Christie. And if you just wait with me for the outro, and thanks everyone for staying with us for this conversation.
Thanks. Thank you.